All right, Stephen, people in the audience are probably asking the question, so what that it's arranged precisely? So what that the sequence is there? If it wasn't there, what would that mean? The cell would be dead, basically, wouldn't it?
Well, right, because the information in the DNA molecule provides the instructions for building new proteins. And the proteins are the toolbox of the cell. They do all the important jobs that keep the cell alive. So if you don't get if you have the wrong sequence of bases in DNA, you won't build a functional protein. When we were together last time, I used a visual aid, and I'll bring it up again if you don't mind. It gets across. These are snap block blocks, but I use them to illustrate how proteins are constructed. Proteins are long chain-like molecules. They're made of smaller chemical subunits called amino acids. Each of the beads here represents one of the different 20 different types of amino acids. If DNA sends a signal, sends information that tells the cell the sequence in which it is to attach one amino acid after another. If the amino acids are attached properly, then the chain will fold into a three-dimensional structure that will have a hand-in-glove fit with other molecules in the cell and will allow the chain to do the job it needs to do. So if you get the sequence right of the bases on the DNA, then you'll get the correct sequence of amino acids, which will cause it to fold properly and which will enable it to perform a function.
This has been an ATRI production.